I'm in the Department of Ecology and Evolutionary Biology, and evolutionary biologists love to start their talks with pictures of like some swath of diversity of the organisms on Earth. And so I am going to do the same. I am very interested in understanding variation in infectious diseases. And a lot of my research seeks to answer the question, why do some parasites cause severe and/or highly infectious disease while others don't? And this is a bit of false advertising. I don't really study this vast diversity of organisms that you see on the screen, ranging from worms to protozoa, bacteria, and viruses. I mostly focus on malaria parasites. So I'm trying to answer a more specific question: why do some malaria parasites cause severe and/or highly infectious disease while others don't? And the way I've phrased this question kind of suggests that maybe there's some sort of like variation across parasite genotypes. And this is a source of variation that I'm really interested in. And basically it's asking the question, like, if you look at these parasites under a microscope, they're basically like morphologically indistinguishable, and yet they can produce really divergent infection dynamics. So this is showing you logged parasite densities over time in experimental infections in lab mice. So these mice are highly inbred. There should be very little variation across individual hosts, and they're housed in the same environment. So controlling all sorts of factors, but altering the parasite genotype, you get pretty different infection dynamics. So each point represents 5 to 10 replicate infections in mice and some standard errors on the means of those points. And you can see that parasite genotypes represented by the different colors are really different. And these patterns are pretty repeatable. So there's differences in the peak infection density, and then there's differences in what happens after the peak, whether the parasites are cleared from the infection or maintain some high densities. So there's a lot of variation according to parasite genetics, which we'd like to understand, but that's just one of many potential sources of variation. So another one that I'm quite interested in is initial infection dose. And if you look across different kinds of infections, you see like wild variation in the infection dose that a host is exposed to. So just a few examples: E. coli, apparently depending on where you get your exposure for E. coli, the dose that you get can vary dramatically across 5 orders of magnitude. Ditto for viruses. Here's some estimates for HIV infections, and also again for malaria infections, where the initial cohort of infected red blood cells can again range really dramatically across many orders of magnitude. And so for malaria infections, going back to these experimental infections in mice, we have some understanding of the consequences of this variation in infectious dose. So what I'm showing you here is, again, dynamics of parasite density over time across infections in mice. Right, so we have this highly controlled system again, and the thing that varies across these different really hard to read lines of varying dashedness is the dose. And so there's kind of like this rule of thumb in experimental rodent malaria infections, where if you increase the dose by an order of magnitude, you reach the peak of the infection about a day earlier. So this outcome of the variation in dose is really well known, but what underlies that outcome, what is driving that difference in outcome, is not so clear. And then a third source of variation, which I think is really interesting, is host genetics. And it's particularly interesting for malaria because in human malaria infections, we have a pretty good understanding of a number of single locus mutations of large effects. So the one that most people will probably be familiar with is the sickle cell trait. So this is the story, again, that you're probably familiar with, where individuals who are heterozygous at this particular locus have a sickle allele and a wild type allele. They're offered this really strong protection against severe infection. And the map is showing you the frequency of this sickle allele in part of the world. A less well-known, but I think also important and at a higher frequency mutation is this Duffy negativity. So this is actually a null allele. Individuals who have a null allele don't produce this antigen receptor, the Duffy antigen receptor, which is an important binding site for a particular malaria parasite species. And this map shows you that this Duffy negativity, this Duffy null allele, is essentially fixed in much of sub-Saharan Africa. So we have good understanding of these sort of like large effect mutations, which confer some resilience to malaria infection in humans. And one paper I'll plug, because I think it's super cool and it's like super recent, a couple of weeks ago. So this paper is looking at subtler variation in red blood cell physiology that seems to have an impact on malaria parasite performance. And anyway, I think this is a really cool paper, which I might come back to at the end of the talk. But going back to, so this is in humans again, but going back to the experimental infections in mice, we can look at this one study that looked at 8 different inbred mouse strains. These mouse strains, again, each individual strain is highly inbred and there shouldn't be much individual variation, but across these 8 strains, they cover about 90% of the genetic diversity that is used in common lab mice. If you give these 8 mouse strains the exact same challenge, the same parasite genotype at the same dose, you get wildly different outcomes. So some of these host genotypes have no problem with the infection. They survive a couple weeks. 100% of them survive for that span. And then other genotypes do really, really badly. Less than 5% of the individuals of this genotype survive this same challenge. So huge variation in response to infection and in resilience to infection, clearly driven by host genotype. But again, the mechanisms underlying that variation are kind of unclear. So in this talk, I'm going to try to address all 3 of these sources of variation and talk about the mathematical models we've developed to try to illuminate the biology that is driving the differences that I've kind of suggested. Okay. So I'm going to give you kind of like the basic biology description, which underlies all of the models that I'm going to talk about today. So we've got these extracellular parasites called merozoites. So we're tracking those and we're tracking host red blood cells, which are the resource for this parasite. Really quickly, these merozoites have to invade a red blood cell or they'll die in the bloodstream. And that happens very, very fast, like on the order of minutes. Within half an hour, they have to find a red blood cell to invade. Once inside a red blood cell, infecting a red blood cell, those parasites are replicating. And eventually they will burst out of that infected cell, releasing the progeny parasites. And that whole process, depending on the parasite species, it takes like a very particular amount of time. And for the rodent malaria parasite that I've mostly worked on, that process takes 24 hours. And it happens roughly synchronously. So all the parasites in all infected cells burst out at roughly the same time. Of course, we have some background red blood cell mortality, which is going to affect uninfected and infected red blood cells both. And then we have some new red blood cell production to account for in this system. So that's the basic model. Everything I'm going to tell you about builds off of this general description of the biology. So I started thinking about parasite genetics so many years ago now when I was a PhD student. I'm going to try to not have an existential crisis during the seminar about how old I have gotten. But anyway, so I was shown this data. So this data came out of Andrew Reed's experimental group. One of the reasons I really liked this data was because this experiment did an immune depletion of the mice. So they gave the mice anti-CD4 antibodies. So the mice had depleted T cells, which should, they have a large role in the immune response to malaria. And as a grad student, I was like terrified of immunity. So I was like, maybe we can model this system completely ignoring immunity. So we wanted to generate a resource limitation model where the only thing that's like governing the parasite dynamics is the red blood cells that they have. So we had these 2 different parasite genotypes, which are known to differ in how virulent they are. So how severe the infections were in the mice. And I'm showing you the data. Again, these are averages across multiple experimental mice. The solid symbols are the red blood cells and the open symbols are the parasite densities over time. And we can like spend some time looking at the differences in dynamics across the genotypes, but that's not important. What you need to know is that the green one is more virulent than the brown one. So we set out with this basic description of the biology, and I built a discrete time model because of this discrete nature of the life cycle of malaria parasites. I actually didn't explicitly track the infected cells. I was explicitly only tracking the merozoites and the red blood cells. And this kind of allowed me to, by assuming that the invasion process was fast, I could find a solution for how many infected cells there were within a given day. I was really interested in testing different biological hypotheses for what governed dynamics here. So I had a few different variants of this model. So in some of them, I included red blood cell age structure. So red blood cells, when they first come out of your bone marrow, are big. They have some intracellular stuff still. And so parasites might treat them as a qualitatively different resource. So we allowed that possibility in these models. And then I had a few different descriptions for how red blood cell production should look. Right, you'll notice there's nothing to do with immunity in this model, because again, I wanted to just have this resource limitation model. And I fit this model separately to individual mouse time series. I did this with a maximum likelihood approach and a brute force parameter search, because that was my approach. That's why. I fit the models only to the red blood cell data. So we have these time series for red blood cells and for parasites, fit them only to red blood cells so that the parasites provide some sort of like additional test of the accuracy of the model. Okay, so I'm going to show you some model fits, some outcomes of the model. This is a single mouse. So the data, again, the data is in these open symbols and the dashed lines, and the best fit model outputs for red blood cell dynamics are shown here in the solid line. Again, the model was not fit to the parasite dynamics. That's part of why these parasite dynamics look so terrible. What I'm going to show you is some different variants of the model structure. So this was our baseline model structure, and then we increased the complexity of the model. I think there was 8 models in total, and this is showing you the best fit of each of those models to the single individual data set. And if you statistically compare the fit of these models to the data, what we found was across every individual, there were 13 individual mice, the same model was chosen as the best one. And this model included a particular description of red blood cell production, and also it suggested that red blood cell age structure was really important. But in terms of differences across parasite genotypes, so what was driving the differences in dynamics we saw across genotypes, there were 2 main differences. One of them was the burst size. So how many progeny parasites were produced per infected cell. And that was higher for the more virulent parasite than for the less virulent parasite. The second thing was differences in invasion rates. So the more virulent parasite had faster invasion rates than the less virulent parasite. And there were differences according to the red blood cell age structure as well. Right, so those two main traits of the parasites seem to vary. Maybe this isn't, maybe these traits aren't like a surprise as the thing that's driving differences in the dynamics, but it's not really something you can tell from just looking at the time series data alone. One thing that we thought was pretty cool is that these genetic differences could predict the outcomes roughly in mixed strain infections. So this is not a model fit. This is taking the median estimate for the 2 different genotypes and essentially putting both parasites in an infection together and then predicting what would happen given initial conditions for a particular mouse. So these are literally just predictions, not model fits. But you can see that it does a pretty good job of capturing what is happening in these mixed strain infections. We're slightly overestimating competitive suppression later on, and that's probably because later on immunity is becoming more important and we've completely ignored immunity. Okay, so we were pretty happy with that that model validation, but we wanted to go farther and sort of like empirically test these quantitative predictions and qualitative predictions about the traits that vary between the 2 genotypes. So again, those 2 traits are higher burst sizes and higher invasion rates in the more virulent parasite. And I worked with Sarah Rees in Edinburgh to do this experiment, and we found evidence to support this difference in burst size. So we counted more progeny parasites in infected cells if it was the green more virulent parasite than the brown less virulent parasite. Invasion rates, we didn't find what we expected. We expected this to be higher again for the green parasite. We also expected the invasion rates of young red blood cells to be an order of magnitude lower, and they weren't. So we were a bit confused about what to make of this. Was it a problem with the model? Was it a problem of letting a theoretician do an experiment? We weren't really sure, but we went back to the model to do another iteration of model development and fitting. So the thing that we added to the model was immune responses. And we added 3 different immune responses, which are independent, which is why they're kind of shaded differently. But we had immune responses that could target infected cells, these extracellular parasites, and uninfected cells as a sort of bystander effect. And then the way that we modeled immunity here was super phenomenological. We imagined that there were sort of windows of immune killing activity, and then we added an additional death rate to those different classes. And the size and shape of that window of immune activity. Right, and the take home is when you allow this in the model, there was a few other things that were different about our process, but when you include these immune responses, you can actually do a much better job of capturing the dynamics of these infections. The parasites are now plotted on a log scale, which makes it hard for you to do the visual comparison, but these fits are way better. So what do we learn from doing this second iteration of model fitting? This is the wrong, that doesn't matter, but this is not the right reference. This should be a 2011 paper, but also in MNAT. What we learned from this second iteration of model fitting was that we found this burst size effect to come out again of the model fitting. So now this prediction that the more virulent parasite has a higher burst size survived two rounds of theoretical examination and 1 round of empirical examination. So we're pretty confident that this is a real difference in parasite traits. The second thing we discovered, which was perhaps more surprising, was that immunity was really important for describing dynamics, but only for the infections with the more virulent parasite. So you didn't really need immunity in the model to capture dynamics for the less virulent parasite. And I think this is interesting in part because of the potential for immunopathology. Like maybe it's something about these immune responses that are actually explaining why the mice seem more sick when they have this more virulent parasite. So if we go back, if we revisit that target cell limited model that I started with and actually plot the dynamics on a log scale, you can kind of see what's going wrong in general, or where the model is doing poorly. And the model is doing poorly by over predicting the density of parasites when you have infections with the more virulent parasite. And if anything, it's maybe under predicting densities for this other parasite genotype. So how do you fix that? Like you can fiddle with resources a little bit, but essentially if you just like slam on some immunity, a window of immune killing that can like absorb those extra parasites and actually fit the data a lot better. Cool. So for parasite genetics, I think we found some evidence that there are differences across genotypes in their replicative capacity. This seems to be mostly driven by burst size variation and not other traits like invasion rates. And then secondly, there are differences in how hosts respond to the different genotypes. So I've showed you evidence of that for immunity. I didn't show you evidence, but also there seems to be differences with respect to the red blood cell production in response to those infections. Again, maybe these are not surprises, but we can like kind of quantify those differences with the modeling. Okay, I'm going to move on to the initial infection dose and the variation generated by that. And this is work that was done with these folks here. So David Schneider is a prof at Stanford. I think he's the head of immunology and microbiology there. Megan was a postdoc in my group who's now an assistant professor at Cornell. And Sakushi was a PhD student in my group who led all this work. He's super amazing. The beautiful graphics are his. He's currently a postdoc in Edinburgh, but soon he will be taking up a fellowship in Paris. Okay, so we saw this variation in infection dynamics across doses. This is an amazing data set. but we can kind of like summarize the consequences of that variation in dose in a couple different ways. So the first one, this is the one I described to you before. If we look across doses, as we increase dose, we basically speed up the pace of infection. So the day of peak parasite density becomes earlier and earlier as you increase the dose. The second thing, the peak parasite density itself also seems to change. You roughly see this increase in peak density as you increase the dose. And going along with that increase in peak density, peak parasite density, is you get a decrease in the minimum red blood cell density. So again, that's the resource for the parasite. So it makes sense, as you increase the peak, you decrease the trough in their resources. So we have these rough patterns that we can describe of the dose dependence in infection outcomes. Of course, you'll notice that there's piles of individual variation as well in these outcomes. And so Tsukushi's plan was to develop a mathematical model that allowed him to dissect both this dose dependence and this individual variation on infection dynamics. And he wanted to develop a new model because he wanted to add more realism to the description of immunity. And the reason for that is because one interpretation that was offered to explain those differences in infection dynamics across doses was that it was dose dependence in immune responses. So we really wanted to kind of see if we could find evidence for that. So Tsukushi took the same basic model structure. He modeled it in continuous time instead, for reasons which have escaped me now. But he used the gamma chain trick with 12 compartments. So that was reducing the variation in the amount of time that parasites spent in infected cells. So you could approximate-ish the sort of discrete life cycle of the parasites. He included two distinct immune responses, which are color-coded helpfully here for you. One which is targeting infected red blood cells. So this green response is just an additional killing rate for infected red blood cells. And then he had this indiscriminate red blood cell killing. I think this is supposed to be a spleen that's filtering some red blood cells, but that will filter both infected and uninfected blood cells. So you see this orangey arrow in both of those classes. So there's slightly more realism to this description of immunity, but it's still like super phenomenological. We're describing clearance rates and we're not talking about particular cells or particular molecules. Right, so this sort of phenomenological approach was inspired by some previous work where essentially what we're doing is we're imagining there's some max immune response, and then we're just like looking at what proportion of that immune response is activated at any one time. So in particular, our interpretation in this one project of this n value, these n values, is that it's the proportion of cells that are cleared per day. So proportion of cells that are cleared per day. This description of immunity is actually quite a bit simpler than what we did before with the windows of activity in terms of the number of parameters that are involved. So that previous description, that was a function of time, there were 8 parameters because there were 2 windows of activity, each window described by 4 parameters. This, we've only got 2 parameters. One of those parameters is an activation strength. So immunity is activated in response to some cue in the within host environment, and that activation happens with a particular strength. And then there's some decay rate. How long do those responses stay active before they stop responding? So we only have these 2 parameters here. We assume that the within host cue that hosts are using to ramp up immunity, whatever that cue is, it's proportional to the abundance of infected cells at any given moment in time. And so in reality, innate immune responses against malaria and many other things are thought to be triggered by PAMPs, pathogen-associated molecular pathways. So we think this is a reasonable assumption to have our within host cue be these infected cells. And in fact, we look at the ratio of infected cells to the maximum number of infected cells observed in any host at any time across the whole experiment. So I realize for a math audience, this is literally the only equation I'm going to show you, but this describes the change in this immune response. So we've got some within host Q, some activation strength that turns on, the chunk of immunity that's not already on, and then we have the decay of that response. So again, the interpretation of N here is the proportion of cells removed per day. So what we do is we convert this into an estimate of a clearance rate in our dynamical model. Okay, so once again, we have a lot of time series data with red blood cells and infected red blood cells from these mice in this experiment. And Tsukushi wanted to simultaneously, unlike what I had done before with these individual fits, he wanted to simultaneously fit across all of this data. So multiple individuals, multiple doses. And so he did this with the hierarchical Bayesian approach. And for anyone who's interested in the details, he did it in STAN through the RSTAN interface. So again, he wanted to capture this individual variation. And he did that using these partially pooled parameters. So a particular parameter for a given mouse is going to depend on some average estimate of that parameter plus some individual deviation. And so those individual deviations are drawn from this normal distribution, and we're actually fitting the variance of that distribution with our model fitting routine. So we're fitting a parameter, a median parameter, and we're fitting the variance describing the distribution of individual variation. And then I think I was really excited about this. So he wanted to describe the dose dependence, and he decided to do this using essentially linear reaction norms. So we have some parameter of interest, and it's going to change in some way with initial infection dose. For the most part, we didn't really have a prediction for which way these things would go. So just one example, thinking about immune responses, the expectation is that you would have increased immune activity with increased dose. Increased threat should generate an increased response. But there's also evidence that higher doses can overwhelm immunity. So we weren't sure which way these things might go, but we allowed there to be for each parameter, we also fitted a slope on that parameter, a slope given the dose. And we also allowed individual deviations in that slope. So for those who are keeping track, I haven't showed you this, but we have 6 biological parameters in this model. Each of those parameters has an intercept and a slope, plus variance, plus a distribution describing the variance around those values, both for the slope and for the intercept. So that is 24 parameters. We've got 51 mice in this experiment, each with infected cells and uninfected cells, and at least 10 days. So we have well over 1,000 data points for those 21 parameters. Okay, apologies for the black background here. Tsukushi makes these beautiful black background talks, which were great in person, but I feel like virtually black background is terrible. But I wasn't able to convert this one. But this is just to hopefully convince you, if you can see it well enough, that the model does a good job of capturing the infection dynamics of red blood cells and infected cells over time and across doses. So what did we infer about dose dependence, the effects of dose from this experiment and model? The first thing is that there isn't really evidence of dose dependence in parasite traits. This was the only source of parasite-specific variation that was allowed across doses. Invasion rates, we found no evidence that those were really variable, at least across genotypes, so we actually just set those in this model. So this was the only fitted parameter of parasites, and it didn't vary by dose, which was sad for me because I had a pet hypothesis here that parasites respond to their environment in terms of number of conspecifics and do something different with their traits. But the fact that this line is dashed tells you that when Tsukushi did an analysis, removing the slope parameters, didn't fit a slope, just fit the intercept. That model fit just as well as the model that had a fitted slope. So no, you can drop dose dependence in this parameter and the fit is fine. He did find evidence of dose dependence in host responses though. So this asterisk here and the solid line tells you that the dynamics are extremely sensitive to dose dependence in this process. So there's no evidence of dose dependence here for targeted red blood cell removal. So this is the activation strength of immunity, but there's strong evidence of dose dependence in activation strength for this general red blood cell removal. So the way to interpret this is that at higher doses, responses seem to be poorly activated, or potentially they're suppressed. We can't say here, we can just say that they're poorly activated. So it seems like this supports the notion that at high infection doses, it perhaps enhances the parasite's ability to evade immunity or suppress immunity or damage host machinery, something like that. So that's activation strength. If we look at the duration of response, so the half-life in the decay term in the description of immunity, we see dose dependence again, and it's going in the opposite direction. So now this tells us that, those responses that were were poorly activated. At high doses, you have these responses that are poorly activated, but they last a long time. And at low doses, you have the opposite effect. They're strongly activated, but they are very short-lived. So as a consequence of these kind of opposing effects, if you look at the outcome on immunity, if you look at the outcome in terms of how much killing is going on by immunity, you actually find the most killing at intermediate doses. So this plot is showing you proportion of red blood cells removed due to immunity. Doses are on the y-axis here, and the data has been standardized so that the peak parasite density happens on day 0. So perhaps conveniently for the mice, the highest immune activity happens around peak parasite density, but the lighter colors means more killing happening, and the highest killing is happening in these intermediate doses. So I think that's kind of cool that you have these opposing effects of dose on different components of immunity. And the way we sort of summarize that is to say that high doses can facilitate some aspects of host responses, increasing the persistence of those responses, while debilitating other aspects like decreasing the activation strength. This suggests, and again, I don't think this is a surprise, that there's separate host machinery for these different host responses, because one was like highly dose dependent. General immune killing was highly dose dependent. Targeted immune killing was not dose dependent. So that seems like there's separate machinery for those two responses. And then finally, like I said, I'm sad about this one. The dose dependence seems driven entirely by host responses and not intrinsic plasticity of the parasites. Parasites don't seem to be altering what they do. Okay, let's talk about host genetics. So I showed you this plot before, which was describing the substantial variation in outcomes of infection across these different mouse genotypes. I'll show you the dynamics, and they're plotted in this funny way, which I love. So on the x-axis here, we have infected red blood cells, so pathogen load. On the y-axis, we have uninfected red blood cells, so the resources again for the parasite. This is essentially like a phase plane diagram. The infections start in the top left, where you have lots of blood cells and very few infected cells. Parasites grow up in the infection, and you follow the trajectories in this clockwise way. I've got the survival up here, and you can see the patterns. The trajectories that are traced across these different mouse genotypes vary pretty dramatically. And plotting the data this way is something that David Schneider, a collaborator who I mentioned before, is something that he did. And when I saw him give a talk, like plotting the data this way for the first time, I was like, why did I never think of doing that? It's like a really satisfying way of thinking about how the infections are changing. And he thinks about this as sort of disease space. So again, parasite burden on the x-axis, host health on the y-axis, some measure of host health. You can track the trajectory through sickness and recovery and then back to health. And roughly what you see is kind of two main trajectories for the different mouse genotypes. Trajectory A in the light gray, the host gets sick, but the host manages to recover and end up back in a healthy space. So the top row is roughly following that sort of trajectory. And then we've got the second trajectory where in the dark gray, where hosts infections progress, and the hosts never manage to, go back up the y-axis, improve their health again. So these are, hosts that don't make it through their infections. And the bottom row is roughly following that trajectory. Okay, so if we wanted to understand the variation in these infection dynamics, could we explain what was going on? So once again, Tsukushi fit his model to this data. There were a few subtle differences in this model, which I'm happy to talk about, but roughly it's the same mathematical framework applied to this data. I'm going to show you distributions of parameter estimates. These are actually strain specific. This is showing you strain specific variation in parameters. So at 0 here along the x-axis, that's the average of that parameter across all hosts and all genotypes. And then what you see is the pooled deviations across individuals within a genotype from that average. And, a couple of things, these are the 6 biological parameters that were fit in this model. A couple of things jump out at you, perhaps, some distributions are really far off from the mean. So here's one where the burst size is substantially lower in this host. I'll come back to that. Yeah, you can look at these distributions and see that there's some variation. I find that a slightly easier way to view these data is this. So Tsukushi plotted, these are the median estimates for individuals in a single bar, and the bars are stacked according to their value. And a few things jump out again, where you see colors that are clustered together, suggesting that there's some very specific, there is a host strain effect. So here, this red and yellow strain that are pretty poor survivors of infection. They have high estimates of red blood cell production upregulation. So when hosts are infected, maybe they produce even more red blood cells than they would normally to try to make up for the deviation, make up for the loss in an infection. So they have very high estimates of that. This dark blue strain, again, over here is the one I probably should have highlighted because it's the one I pointed out before, where it's got the lowest estimates of burst sizes, but it also has a sort of low activation rate of indiscriminate responses. So we spent some time looking at these stacked plots and trying to draw inferences about genotypes. And then Tsukushi had the good idea of taking this 6-dimensional data and trying to distill it into key functional motifs. What is really driving the variation across these genotypes? So he did a principal components analysis on the estimated parameter set. And you can see the variation explained by these 2 axes. Collectively, they explain about 67% of the variation in parameters. And there's some really interesting inferences. So there's kind of 2 different ways to do badly in infections. So the yellow and the red genotype over here, again, the red genotype has 5% survival from this infection. And what you can see from this PCA is that they have low activation of targeted immune responses. So that's this vector here. That's our targeted immune response activation. They're way on the lowest end of that. So they're doing a bad job of activating specific immunity to kill infected cells. But additionally, they're on the far end of these other 2 vectors here, which are the parameters that govern red blood cell production, both background red blood cell production and then potentially upregulation during infections. That's what those 2 parameters are. So these mice are pumping out red blood cells, which are the resource for the parasite, fueling parasite growth, and then not activating immune responses that would kill those parasites. So that seems like a bad way to survive an infection. Another bad way to survive infection, apparently, is to massively upregulate all of your immunity. So this orange strain, which also was a poorly resilient strain, has the highest activation of indiscriminate responses. So highest activation of immunity that will kill infected, but also uninfected red blood cells. And also has the second highest, maybe not second highest, also has a relatively high activation of these targeted responses. We interpreted this as a scorched earth strategy, or maybe if you prefer sort of immune brinkmanship, just throw everything at the infection and try to kill everything. But in the meanwhile, also kill a whole bunch of your own infected red blood cells to the point that you can't survive as a host. So that's our interpretation of the two different ways to kind of succumb to infection, at least across these eight host genotypes. One, too little immunity. The other one, way too much immunity and not specific enough. It's killing a whole bunch of uninfected cells. If we move a little bit away from these orange points to this highly resilient strain, this light blue strain, we see, a way in which the strains seem to survive infections. So they have slightly less activation of this indiscriminate response and slightly more activation of this targeted response. So this balance in immunity seems really important, not just indiscriminate. You also need a strong targeted response. What else did I want to say about that? This strain also has slightly higher, this is a mortality rate of red blood cells. So it's also got a higher background rate of removing red blood cells. This strain is really interesting. So I want to point it out. I pointed it out to you before on those other plots. It's essentially the Mary Poppins strain, like practically perfect in every way of fighting infection. It's got the highest activation of targeted responses. It's got a high background mortality rate of red blood cells. But the thing I find most interesting is this really low burst size. So that's this beta here. It's got the lowest estimates of parasite burst size. And in part, this makes me uncomfortable because I like to think of burst sizes as an intrinsic trait of the parasite. And this tells me that actually the host physiology can also influence the burst size. But maybe that makes sense. Given that paper I showed you before, that recent paper that shows subtle variation in red blood cell physiology can impact parasite performance, this kind of goes along with that. One element of parasite performance is burst sizes. It turns out this strain of mouse, is known to be difficult for counting red blood cells using flow cytometry, a particular marker that's often used for flow cytometry for most red blood cells doesn't work for this strain. So that, is evidence that there are different receptors on these red blood cells, and perhaps that's what's influencing, parasite success with those red blood cells. Okay, so roughly summarizing that, we think there's kind of two ways. The model suggests there's two ways to succumb to infection: not enough immunity. The poorly resilient strains either have not enough immunity, weakened activation of targeted responses, or too much immunity, really strong activation of indiscriminate responses. And there's also two ways to survive. You want this balanced immunity, a balance in those two different responses, or you want to be the Mary Poppins strain that does everything well. Right, so it's perhaps not a surprise again that the immune system needs to balance removing parasites from the system, but also not harming the self, harming the host. We were interested in sort of linking these functional motifs, if you will, to variation in molecular responses in the host. And so working with David Schneider's group, so Nicole was a PhD student in David Schneider's group. She did a whole bunch of immune assays to look at pro-inflammatory cytokines, which impact malaria clearance, and anti-inflammatory cytokines, which are crucial for limiting inflammation in immunopathology. So she did this. She looked at sort of expression patterns of those different cytokines through destructive sampling of a subset of individuals each day. So this comes from a different experiment. So what I'm showing you here is two different pro-inflammatory cytokines, two different anti-inflammatory cytokines, essentially expression dynamics over time, over the course, over time. The left panel, these are uninfected mice, so showing that there's some variation in expression patterns in the absence of infection. And then on the right, you can see what happens over the course of an infection. There's a lot of points on these plots, and it's a bit hard. It's a bit hard to see them all, but roughly we think that the resilient strains tend to have high expression of both pro and anti-inflammatory cytokines. So the sort of light blue points tend to be high across all of these plots. So that's the sort of balanced expression of pro and anti-inflammatory cytokines. The less resilient strains, there's a couple different patterns. The red strain seems to be relatively low across the board. And then some of the other less resilient strains have really high pro-inflammatory cytokine expression, but relatively low anti-inflammatory cytokine expression. So Sakushi plotted this also as a sort of ratio of pro and anti-inflammatory cytokines, and you can see that one effect where the yellow and the orange tend to be pretty high. So they're sort of overexpressing pro-inflammatory cytokines and don't have this like concomitant increase in anti-inflammatory cytokines that would, reduce the potential damage of those immune responses. So this cytokine data, we weren't thinking of it as like a model validation thing, but we wanted to look at what the model said were the sort of ecological explanations of what was changing in these infections, and then compare that to the sort of molecular details of what was changing in infections. So I think what we saw was better host survival is associated, from the model, what we saw was that better host survival was associated with these precisely targeted robust immune responses. So that targeted response that's killing just infected red blood cells, you want a lot of that to survive an infection. The molecular data suggests that you want a balance in pro-inflammatory and anti-inflammatory cytokines. You want this well-regulated inflammatory response to survive infections. So as I was putting all of this together and thinking about what we learned about parasite genotypes and host genotypes and infection doses, I started to wonder, like, do we actually understand stuff? And to some extent, I think yes. This audience probably, I'm preaching to the choir here when I say that models are great. They can reveal biology that's not obvious from looking at the data alone. And combining that with new experimental data, I think we really have generated new understanding of some of what underlies the variation in infection dynamics, some of the mechanisms that give rise to different outcomes across these different sources of variation. I also think, my group, we're pretty happy with the structure of the model we've developed, including this Q-based host immune responses. Part of why we started these projects, we were trying to do a completely different project where we wanted to understand variation in responses to drug treatment. And Sakushi said to me, what model should I use as the sort of base model on which to apply drug treatment? And I was like, I'm not sure. Like there's a bunch of models out there. I don't know which one's great. And so we decided to develop another one. And through these two projects, we're pretty happy with that model. So now I think we have a baseline model that we can use in other situations. The challenge, of course, is that we've only scratched the surface of variation. There's lots of other sources of variation, more than just these four even. It's variation in host condition, variation in previous exposure to infection, which is going to influence all of these immune responses, variation in interventions, and of course, co-infections with other parasites. That last one is something we've worked a little bit on. We had a recent paper where we were trying to understand variation in responses to infection with and without a worm co-infecting and across different doses of malaria parasites. And so we've gone some way to explain that as well. But I think generally what is true is that there are no generalities here. Those probably don't exist, and it's probably unhelpful to think that they might. We can generate understanding of what's going on in certain conditions, but prediction in other conditions is going to be really hard. And I think that's probably important to know, especially for, important things like infections. So I will stop there. Here's all the folks that I worked with on those projects and some folks who funded those projects. Happy to take any questions if you have some.
Thank you, Nicole. I have so many questions, but I'm not going to be the typical organizer who just totally monopolizes, but I am going to take the first question because
okay.
So I thought this was such a nice talk because I'm also interested in these kinds of levels of heterogeneity and what drives what. And I think what's interesting is in some of the work that we've done, we found similar things through completely disparate modeling strategies, particularly things like this scorched earth pattern where you get complete activation or very fast activation of macrophages that then kind of dominate the immune response in a kind of non-targeted way. And I was interested also in this activation, strength, and duration of response, where you saw this, like you had those graphs that went in opposite ways.
Yeah.
I wondered if you ever thought about using like the area under the curve as some proxy, because that would give you some idea of exposure. And maybe there's another part of that sweet spot in the intermediate that has to do with exposure.
So my gut reaction, which tends to be very false, is that I wonder if the area, what you're thinking for an area under a curve approach would be different than what I showed with like the functional outcome of those varying responses. So if we look at, we've got this activation strength, we've got this duration of response, that like the consequence of those depends also on the dynamics, because the abundance of infected cells is going to impact what that activation strength actually does. And right, so when you put that all together, you get that heat map that I showed. Yeah, so that those are my initial ramblings. I'm not sure if you're thinking of something like fundamentally different than what that plot shows, which is essentially the consequence of the
thing. Yeah, no, I was just thinking if there was like quantitative, like maybe you could I don't know, like, again, this is my gut, I just like go in all directions. So, and I also have been feeling really old recently. I laughed several times during your talk because I just felt like I was listening to myself in my own thoughts. Because I've also seen David Schneider's talk and I loved that graph that he had. And I was just like, yes, this is what I thought as well. I was just thinking that you could perhaps find a kind of sweet spot, like a quantitative sweet spot, where if you compare the AUCs of both of these responses in whatever metric, in whatever dynamics you had, that you could find not a separatrix, but like a kind of region in the space that gives you some idea about what the optimal balance, or like where the optimal balance happens to live.
Yeah, one of the things that I often get asked about, and one of the things I often get asked about is like, where are hosts doing the right thing? Like with the dose experiment too, like, are they responding the best way to those different doses, or could they be doing better? And it's odd that, so much of my time I spend doing like evolutionary projects, but I haven't like combined the two to be like, are these host responses actually the best that a host can do, or could they do something better? So that's kind of along the lines of what you're saying, like what's the optimal balance? I think there's a bunch of work we could do there for sure.
Yeah, and I think, just personally, because I was talking to someone this week who said, it's kind of controversial to say that the response to inoculation is in some way not dose independent, but like also, and we also find this, that like, yes, of course, you're going to have some increased exposure is going to increase severity in a large cohort of people, but those who are going to have a severe response are going to be the most severe across all of these inoculations. And I think that that's an important point to think about. It's not just, like it's exactly what you were saying in the entire talk. It's this whole, combination of all of these factors that contribute to heterogeneity. So I really enjoyed it. I'm not sure if there are any other questions from anybody else, even though I just kind of dominated that section.
I have some question too, but if some other people have another question before me, they can ask. Well, you talked about windows of immune response, and you said they were not really like physiologically based, or like in your reasoning on why you applied them. But I was just wondering, why did you apply them?
Yeah, I should say they weren't really physiologically based, but I think why we applied them was kind of inspired by my limited understanding of immunity. So that limited understanding is that early on you'll have some innate response. And later on in the infection, you're going to get an increase in other kinds of responses that actually like do the job of clearing the infection. So I think that's why we were thinking of these sort of two discrete windows of immune killing. But they're not physiologically based in that, they're literally written as functions of time and not, something physiological.
Yeah, okay, I get it. And also, you talked a lot about these plots there with that are it's the number of red blood cells and the infected, the curve. I know some of them had like a 72% survival, right? But I didn't really see like a like the curve wouldn't go back to recovery. So I was wondering if these data in particular, they all didn't recover.
That's a great question. I don't immediately know off the top of my head whether I don't immediately know off the top of my head whether the plots I showed you end like if they're ending at a particular day and survival is to some other day. It's a great question, which I cannot answer off the top of my head, which is really embarrassing.
It's fine. If ever you know the answer, you can email me. You have my email. So
yeah, I mean, it could be like, yeah, it could be that there are like a lot of lines that we can't really tell are ending early in that plot. I think that might be what's going on. Like some of them, I'm sorry, it's not fair that I'm looking at this plot and you're not. I could make that better by sharing. Like for this red one, you can tell that some of the lines are really short. So obviously that's a host that is not surviving. And I can't tell you from looking at this which host is surviving. If there's like a single host in here who's surviving, who is it? It's probably this one because it's very long. Your point about them not going back up to recovery, that is, over this time scale, they don't go back up to recovery. But I've actually seen those immune depleted mice that I talked about in the first project. We maintained those infections for a really long time to try to see if my estimates of what should happen in the long term would be true. Those mice had like massive parasite densities, but were like still alive 6 weeks later. And I think that was because of the advantages of the immune depletion. So it's a slightly different story, but you can have like relatively high densities for a long time and still somehow survive that infection.
Okay, well, cool. Very nice talk. I really like the PCR analysis too.
Yeah, cool. Thanks. Me too. Not my idea.
Great. Well, we're at time, so we'll end it there. And thank you again, Nicole. Such a great talk. I learned a lot. Thanks very much.
Awesome. Thanks.
Thanks, everyone.